just under 2,000 meters. It's hard to imagine how a storm could have that.
Okay, I'm going to black balance and it's going to go black for a minute.
Effect.
Roger. Can I, actually, sorry, can I keep bubble on the front there?
So as most of our viewers know, what they're doing now is calibrating the camera and the white balance on the camera at the depth and color of the ocean that we're at. And that's important for getting correct color for the organisms we're going to image.
Thank you.
If you don't white balance, what you get is a lot of blue tinged and off color, not the right amount of red, particularly for the coral cameras. We have the shallow water one, so it's important to balance that. You have to do it every time because your conditions are always changing at the depths that you're going to, the location that you're in.
Okay. It's looking good.
SPL's not looking great.
Nope. You need to do something?
No, not yet, but if it doesn't straighten itself out, maybe at some point.
Okay, I don't know why. That just started.
Yeah, no. I doubt you're to blame. You want to go?
Say again?
Are you ready to go?
I'm wondering if that tripped it into transponder or something? Because of this is power cycle?
Oh.
Is there a way you can query that?
I don't know. I can look on sonar. Well, I mean, Sonardyne was still going. I kept seeing stuff come in.
Okay.
I can scroll through fixes.
Can I grab bubble for a quick gauge check?
Yeah, go ahead.
I'm getting continuous fixes, so I don't think so.
Cool. Oh, we just did it, and then it fixed itself?
No, I don't think it power cycles. And I'm not ready to power cycle it because it's not too bad yet, but if we get more of this.
Okay.
Oh, well, sorry, I thought you were saying that you thought it might have power cycled when you restarted the.
Oh, I was thinking it might have done some weird setting.
Oh,
who knows?
I don't think so, but I don't know for sure.
Okay. Cool. As soon as Antonella's done her gauge checks, we could do our start ceremony and then be on our way.
I'm ready.
Okay, great.
Are we ready?
Yeah, we're ready.
Hey.
Okay, great. So for all of our viewers following us at nautiluslive.org, we have arrived to Don Quixote Seamount Northwest, and we are going to go radio silent to conduct a private ceremonial protocol to acknowledge and honor the biocultural resources that are present and that we are interacting with and impacting. So we'll be back shortly. Thank you.
All right, back row, you guys ready?
We're ready.
All right.
We're ready.
All right, you want to just start going up the ridge?
Yeah, let's take a look at, since we have some animals on the bottom from the get-go, let's take a look at what we've got growing here.
Okay.
Sure.
So how about that sort of fork bamboo to get started?
Forked
That's
bamboo,
directly
sure.
in the center of the camera. Wait, I'm going to use my toy. Here we go, right there. What's my toy called again?
Telestrator.
Telestrator.
Yeah, I love that term.
And
Okay, zoom in, please.
I have a note from the previous data logger. I'm not sure if it was started, but if we could start the still cam.
Where does that get started?
It just says when we reach the bottom.
I think I have control of that, but
Do you? Okay.
I think I can tap into it.
Okay. I turned the power on at the surface, so it's out of my hands.
Thank you guys.
Well, Chris, what do we got here?
This is a bamboo coral, but could you look at the one right behind it, the smaller one?
Yeah,
You have a chance?
I think so.
Okay, I do not have it, control of it.
It's going to be right up a little bit, right there. Right, that guy right
All right, Aaron, you got some zoom for me?
Yeah, I do.
I'm really far away.
I do not have any more room for you. That's it.
So the reason I'm looking at this particular bamboo is we do have a request in for one of these sparsely branched bamboos, and we've seen some, but they've all been too big because it's very important to get the nodes or the joints where the branches come
I
out.
see.
I'm wondering if this is small enough where this would actually
Come wide, please.
work.
Yep.
Do you think you could actually get most of the specimen? Because or is it still a little bit too big to kind of fold into a box?
Well, I think we could fold it into a box. So make sure I understand the concept. The goal is to gather the nodes, the branching area.
Yeah, and the problem like this one in front is in order to get that branching area, you've got to take so much of the top that it's a very difficult collection, but that one in the back is smaller.
Is smaller. Well, the other option available would be we could snip off the top and then take the bottom.
I'm not sure I understand how that would work. Sorry,
So
frog in my throat.
Yeah, imagine this branch. So on the one you're talking, just to the right of the lasers now.
Right.
See the big long branch on the left side of it?
Oh.
We could coral cutter that and drop the top, leave it behind, and take the rest of it.
I think that's a good plan. I think
Want to try
that
that?
could work.
We could, I don't know how brittle this is, but if it's flexible and kind of bendy, we could even slurp it.
It's flexible.
Okay, we want to try that?
Yes, absolutely.
As long as I put it base first.
Right. It's the first one I've seen that even offers an opportunity to fulfill that request.
Okay, I'll see if I can, how am I going to squeeze in here without landing on other corals? That's going to be the problem here. I think this side's going to be the best bet.
So a viewer wants to know if we cut, if we leave part of the coral behind, would it keep growing?
Yes, the answer is yes, it would. But under these circumstances, there's obviously some other colonies close by of that same species, we believe. And this one right in front of us, this larger one, I believe, is an older one of the same that's behind it. And because it's so important for them to get the branch points on there, we would be taking most of the sponge and leaving very little tissue on it. So
it just says ask the pilot or video engineer. Sorry, did you hear that, Aaron? Okay. It says pilot or video engineer.
For the camera? Yeah, camera's powered on. That's all I've got.
Still cam, yeah.
That's all I can do.
Thank you guys.
Yeah, it's on a specific. Okay.
So before we sample this, could we zoom one more time, do a couple of them? One of them would be on the whole colony that's as close as possible, showing the whole thing. And
Yeah,
then
roger.
secondly, a
Stand
close-up
by.
right at the branch points as well.
And you stand by too. I'm going to redo this. Get this a little more stable here. That's better. Okay, you can go ahead and get the arm out, and before we sample it, we'll get a zoom in on it.
Okay, thank you.
Yeah, roger. We'll get the manip out first. We might be able to slurping the whole thing will be too long. That won't work. Okay, then just get that off to the right side there, and then we'll do a zoom.
Sorry, Antonella, it's the one in the back there. If you could move the arm a little bit more to the right,
she's
we're looking
working
for
on
that small one. Oh, okay, I'm sorry. I was thinking she was targeting that big one. But thank you.
Yeah, so just get the arm out of the way for our zoom.
Oh, sorry. Okay.
As soon as the arm is out of the way, yeah. That's good there.
So first, just the whole colony is tight where you can get all of the
Okay, go ahead and
branches
zoom.
as close as possible. And maybe back out a little bit, again, just to get the whole colony. Right about there. That's really nice. Hold it for a little bit.
I'm going to get rid of your lasers.
Okay, and then if you could zoom maybe on that close branch there so we can get a look at the polyps and the branch point.
You okay with another reset?
That's all the zoom I
Yes.
have.
Okay, thank you. Okay, I think that'll do it. So I thank you very much. Appreciate it.
Yep.
So that left branch, we'll snip that off first.
Okay.
Have to turn the coral cutters around 180.
Perfect.
Before you get in there, turn the coral cutters around. There you go. Yeah.
That way.
Speaking of black corals, there's one right behind it.
Yeah, a little
Bathopathes.
Bathopathes or something. Meredith Everett had noted also there's a mushroom coral right at the base of the big bamboo.
Yeah.
So like, how much do you want me to snip off on this?
Heteropolyp. What is it?
Just like
Heteropolypus.
Heteropolypus.
Getting better at saying that.
I like that. Makes me think of platypus for some reason.
all this.
Is that helping or making it worse?
Heteropolypus.
Silly
It's-
bar.
Base of
I know,
bamboo.
right? It's always in the worst spot.
Another one for you is if you wrist left, you can get that forearm out of the way. You know what I mean? Move the whole thing like that. Yeah, exactly. Keep that going. Then you can see what you're doing. Yeah, there you go.
Okay.
And
So
you call
just-
for zooms as you need them.
Yeah, can I get a half zoom? Yeah, perfect. Yeah, keep it there. So just like
Just
10
clamp it all
centimeters
the
off of
way.
this, or?
Yeah. You can go down closer to the node. Oh, that's fine. Yep, good. Now give it a little tiny twist with the wrist. A little bit, tiny bit of wrist rotate. Good. And then now you can pull it away.
Okay.
Oh, maybe more twist of the wrist. That's not
It's
snipping,
not in.
is it?
Hang on. Let me
Try
read.
grabbing it again. Try just snipping it
Yep.
again. It might be just at the tip of the jaws instead of in the knife part, so I need to redo it again.
Happening.
Yeah, I think it's, you got to go in farther. There you go. Maybe not yet, no.
Hang on.
Kainalu.
Oh, sorry. What was that?
That's it. Yeah, you got it there.
Sorry, I'm not sure.
Good.
What
Great.
would I be?
Now you can let that go.
Do I? Sorry.
Or we can slurp that. No, it doesn't matter.
Oh.
No.
Yeah, I have the regular.
Okay, now get the arm out of the way and we'll assess the plan.
Yeah.
Okay.
Yeah. Okay, thank you.
Aaron, can you zoom wide? Thank you.
Yeah, good there. You can hold the arm there. So I think we want to grab, again, the same orientation of the coral cutters, come in below everything, grab that branch.
Okay.
And
bring it back.
Bring it back. And we're going to feed it bottom first so that the arms of the coral fold in.
Okay.
You know what I mean?
Yep.
As it goes up the slurp tube.
Right. Okay.
So yeah, go in at your leisure. And once you got it, we'll assess the next steps.
Can I get a half zoom, Aaron?
Sorry. Yeah, nice. Pushing the vehicle.
Oh, sorry. It's halted. It's my bad. Any chance you could move bubble up a tiny bit?
There you go. Nice. Great. Now just lift straight up. Perfect. Okay. If you line it up in front of that suction hose, you should be able to stay in place. I'll follow the arm down. So this is going to go into sample jar 1.
Sample 1. Thank you.
I mean, it's set up. I got it.
Say again?
Nothing.
Okay. This might end up being a multi-grab. It might not line up perfectly, but we'll see what happens.
Can you turn the valve on?
Yeah, sure. There's 40%.
Okay. I'm going to open it. Yep.
All right, you can just re-grab it.
Okay.
And try and grab it with the tips of the jaws.
Okay.
Even right there would be fine. Oh.
Oh.
Sorry, I shouldn't say oop. That's not helpful.
What's it? It's not.
If you need, we could reposition the vehicle. Let's see if you can get it there first.
Is it worth just backing up and just grabbing the slurp hose and?
Probably better to feed it in.
Okay.
That way we can control the angle of it.
It's right on the. Let me just bring it up a bit.
You can see it in Hercules too.
What's this one?
Oh, okay. Keep missing it.
Oh.
Damn it.
I'm going to get out of there.
Sorry. Yeah, if you can.
Okay, pull the arm back,
reposition.
please.
Yep.
Wait for the dust to clear. We'll come back. We'll grab it. There's still current here, which is nice, which means dust will clear and we'll come right back. Your regularly scheduled programming will be back in a moment. You see it?
All right,
Just above the...
Yeah.
Yeah. That's stable. That looks stable. Go ahead. Remember, use all the tools available to you, including Aaron Rainey.
Yeah. Can I get a half
I'm
zoom?
a tool.
I'm a tool. That's not what I meant. I'm sorry. Now, if you can grab it farther up, you can actually stick the end that you've got into the hose before letting go. You follow me?
Yep, I do. Let me regrab.
You might, I can't tell if it's sticking out right now or not. If it's sticking out, you're good to go.
Let me just rotate really carefully.
There's a fish right above Hercules for the back row to look at. It just swam over.
Grab it.
There
Nice.
it is. Goodbye, fish.
Oh, that looks pretty big. That fish.
It's right in that little groove.
It's
Really making this
perfectly
difficult.
located.
It found the local minimum. Minimum. There's a gap here you can get into.
Oh. Point that at again. What was it again? Thank you. Trevor, can you point to what you said again?
So you're running into this rock,
Yep.
try to go beside it.
Ah, okay.
Yeah, right in there, exactly. There's potential there, yeah. Yeah, good. Nice. Lovely.
Okay. Zoom wide.
Once you get the end of that into the hose, I'll turn it on. It's going to present its own problems because it's kind of sticking up into your armpit.
Yep.
Not trivial, but I think you'll get there. Watch bubble cam. You got the right idea. If you want, sure. Sure. You have the right idea there. It's just gonna take a lot of shoulder down. And once you get close, can you zoom in, please, Aaron? So you had the totally right stance, and then with the shoulder down, it gets you there. You can do a slow kind of rotate and roll, shoulder down combo. And I don't think I'm actually going to be able to stab the thing into the hose, but we can get it close. Oh, bubble, come on. Really? Okay, you can turn the suction on now. Hopefully I can. 40, sure. Just get it started and then. Oh, almost. There we go. And hopefully that'll just go. Huh, just go, I say. Come on. Get there. All right, let's get more percent. Maybe 70. How bendy is this, Chris? They're going to go? Now's the last chance to bail out. I don't think it's going to go.
Can we put this in a box maybe?
Yeah, sure. Yeah, the slurp's working. I can see the flow through the jar, but it's not going to suck it through. I don't think it's quite, I think it's too grippy.
Well, let's go ahead and pull it out so it doesn't clog up the...
Okay, 0%, please.
Filling section.
Let's grab that better.
The actual person's, Dr. Scott France, is on the line right
Come
now.
wide, please.
And he's watching this. So he thinks this is the one that he was looking for. Tried slurping this, Scott. Now we're gonna, it didn't quite work, so we're gonna try to put this in one of the side boxes.
I'm gonna switch your camera.
Stand by. Oh yes, you can, yeah, you can switch the cameras. Whoop, whoop. Okay, Salvo. That was very grippy in there. I was not expecting that. I was expecting it to be a lot softer. Okay, box out, please. Okay, that's in Delta D.
Box D, got it, thank you. And that was sample number 073.
Okay, I'll take forward view, please.
Can you repeat the sample number, sorry?
Oh yeah, 073.
070?
3, sorry.
073.
Yep.
All right, copy, thank you.
Yep.
Okay, boxes are closed. I can turn this one on and come off bottom. Okay. We got there.
Okay. Let's see. Why don't we make some distance here?
Sure. So do you want us to stay on the northeast or southwest side of this ridge?
Let's take a look and again do the old follow the corals bit. See which one is...
Okay, we'll head straight up slope then for now.
Okay.
Which is probably 180, I think.
Close, yeah, sure, sounds good. You ready for a move?
Yes.
You want to start with 20 meters or bigger?
Let's start with 20 meters. I think it might be 165.
165,
I revised my original.
165, 20 meters. Bridge nav. Good morning. Can we get a 20 meter step bearing 165? Thank you.
Like we've got some other species of bamboo there over on the left side, a little more bushy ones. Looks like a couple of bushy ones.
These little stalks coming into the center top there.
Yeah, these colonies that here on the left here, those are bamboos as well, but they're different species. They have a lot more branches to them.
Oh, really? Oh, I
Yeah.
didn't know that.
This is actually a good start for this dive. We've got a fair number of animals on the bottom, so it bodes well for how this is going to go today.
Couple sponges over here, looks like.
Yeah, it kind of looks like the same beast that we had collected before, but we can do some snap zooms just to confirm.
Yeah, you bet. Okay, zoom in there, please.
Well,
It's interesting. It's attached to the bottom. It's non-stalked.
yeah,
I don't see any grappling hook feet either.
yeah.
I don't see what I see.
Oh, it's attached to the rock there.
Oh, wow, on the right.
Yeah.
Well, we just spent a fair amount of time collecting, so I'm a little bit reluctant to try to go for it again. This is an interesting sponge.
We can do a quick sample here, no problem.
It won't take that long, will it? It was.
Don't think so.
Bridge nav, hold position.
Well, yeah, this is
Come wide, please.
this is going to be a very brittle sponge, and the only reason I'm thinking of this is it possible to grab the rock that the sponge is on?
As long as it's not attached to Mother Earth, then sure.
Okay, because I think it's going to be one of these that's fairly fragile. That's the
Excuse
thing.
me, coral rock. Stop that. Yeah, that would be a great idea. We can. You come down to Delta, please. That should be good there. Oop, 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 oop.
Also, we have a rock request, and I see one to the left of that sponge too before we move off, if we get the sponge in the box.
Cool.
That
Okay,
would work.
zoom in there, please. Good there. Are you talking about this rock?
We're a little zoomed in, but I think that's right.
You could even do this rock here. Any rock.
He's looking for one that's about 15 centimeters, so it looked like that one to the left might be about that size.
Now, if they're going to share a box, I would like to put the rock in first so we don't smash this sponge.
I
wanna
don't
put
know
them
if
in
they
different-
should share a box, the rock and the sponge. I
Okay.
mean, the sponge is on a rock, but
Sure. No,
I think the sponge ought to have its own
sounds good.
spot.
See what we got here. What are we working with? Who's that little guy? Okay, perfect. See if
So this
okay,
is
zoom in, please.
in one of two families. This is either a euretid or it's something that we call, well, actually I'm sure it's one of these. They're not even sure what family if it's this other thing. okay.
You can put it on the starboard side there. Boat cam on the starboard side.
Just keep in mind that if
for
the
the
sponge touches anything, it's going to fragment. So it's a very careful collection.
Nalo, did you get some stills on that one?
Okay, come
Yeah, I
wide,
got
please.
Thank you.
Can you tip up, bubble a bit?
Such a beautiful
Just up.
specimen.
Good. One more. Yeah, right there. I'm going to try and do this really slowly and carefully.
Yeah,
You can see how fragile it is.
right. It's fragile. I'll almost guarantee it.
Is this one of these heterote sponges, Chris?
Excuse me.
So like this one, this
That's a possibility, and it
heterote.
also could be in the uretid family. Could be a smaller version of this one that's been problematic.
You can typically tell by the size of the pores.
Yeah, and this heterorite is also one of these that's undescribed as well.
Okay, sample salvo, please. And stand by. Do not open the box yet. Okay, you can open the box now. We'll go all the way. We'll put that in Charlie.
Think I'll fit in one of the small ones, maybe?
Yeah, I think so.
Okay.
Come on, halt, halt, halt. Button is obnoxious. Oop, no, I don't want to go that way. I'm just going to drop it.
Yeah, that looks good.
Oh, I think it detached from the rock.
That's okay as long as
No,
it...
don't float away. Come on, we've come this far on our journey. Oh no, are you kidding me? I got to stuff now.
That's okay. That's all right.
Okay, close that box.
That'll work. That's perfect. Whoa.
Come on, that was absurd.
You did a great job getting it there.
Okay, so let's go for the
That was
rock, and
breathtaking.
the one we're looking at is this guy right there.
Nice rock.
It's about that.
Roger.
That's
And then we also have a request for a water sample, so we're going to do a threefer here.
Threefer.
Threefer.
Box C, just to double check.
That was box Charlie, correct.
Right on, thank you.
Let's take a look at this rock before it goes in
Sure.
real quick.
Let me even put some lasers on it.
I want to make sure it's thick enough if it's too thin.
Zoom in, please.
Oh, I'm sorry. That's not the rock. The one is over to the left.
Okay,
I
come wide, please.
apologize. I think if that's not too big, this one I just circled might be
Yeah, Roger. I moved your circle for you. Boom. It's too big. It's part of the planet.
Okay, how about down below to the left? Let's see.
This one?
Yeah, one of those in there.
It's a terrible grip.
Oops, that's crusty, I think.
Crusty?
Yeah,
Don't
it's
want
too thin.
Okay. Now what?
How about the next one that was over? This one here.
Roger.
Work.
That's angular. Go ahead and zoom,
Yeah.
please.
Let's see if, yeah, let's go ahead with that one.
Okay. Come wide, please.
Any luck? That's not just crust. It's got some basalt in there.
Ooh.
Then we'll get water, and then
I'm gonna re-grab
we're
that.
going to need to
That's,
do some community
my best.
surveying.
Community surveying.
Community surveying. We're going to do a survey of the community.
Okay.
Like the census?
Kinda
Yeah, like the census.
Okay, sample, please. And now we get to see if this is Pandora's box.
So will the
Do you want a big
sponge
box or a small box?
You can put it in the big, and then we'll put a couple in the big one, because you
Big
only have
box,
to
okay,
slide
open.
that out part way, right?
Correct.
Yeah, let's go with that. Keep the sponge from drifting.
Woo, don't do that.
Well, either one, yeah, would work.
Foxtrot.
Great, thank you very much.
Foxtrot. Got it.
Kainala, you saw which one that went into? Great, thank you.
I did, thank you.
I get Niskin cam, please, and dive salvo. Aaron, just want to show you this dive salvo. Now I'll do the real dive salvo. It did the butt cam there.
Sorry, what was that?
That was when we did the dive salvo. I put butt cam where
Before or now?
just now, and then I
Oh.
changed it again.
Okay.
You got it.
Perfect. Okay, well, let's see. We got water, we got rocks, we got a sponge, we got coral.
We've got it all. We can recover.
Yeah, that's it. Nothing to do.
We need to slurp a fish.
Oh yeah,
That
we'll
was
start with
the
fish.
skin one.
So the samples look right, 74, 75, 76.
Yes.
Thank you.
Yeah.
Ooh, look at this falling everywhere. Okay.
And that last Niskin was
Okay.
was it Niskin
Now we're doing
one?
our census.
Yeah, this is kind of sampling for the deep sea coral program to give them some distance here to see what the community as a whole looks like. Maybe we'll do some snap zooms on the way as we can.
We'll start a ship move then.
Yeah, that would be great.
All right, back to 165. Yeah,
Roger.
bridge nav. 20 meters, bearing 165.
Right, and we're off.
Okay, it's another one of those glass sponges. You read it. There was a Candidella gigantea that was on the bottom, and there was a xenometrid crinoid that we were so focused on the collection. One of these sea lilies that was attached to
Oh, neat.
one of them that was really beautiful.
And are these all dead corals that we're looking at? Or are
Yeah,
they-
yeah, red one. So this is really nice to start off with. This could
they're
develop
dead.
into a very nice community.
Yeah, this is great.
Those are branching
Yeah.
Isidae. Right,
One of
bamboos.
our viewers is wondering if the increase in life is due to more optimal currents not being blocked by the seamount.
And you can tell your viewer that I believe so. I think he's absolutely right. But we're not sure, of course, but we think so.
Yeah, thank you for the question and the answer.
What's the one on the top right corner there?
Top right corner.
Coral.
Oh,
Very
well, can we
linear
do a snap zoom?
Yeah, you bet.
Take a look.
Go ahead and zoom there, please. Now that I'm at a terrible angle to see it, perfect. Sorry
Yeah.
about that.
So this is a primnoa. Thank you for pointing that out. This is probably the first one of these that we've seen on this cruise. It's called Calyptrophora angularis, I believe. And I think it's one of the first of that genus that we have imaged. And what am I looking at? Well, angularis tends to have sort of these two branches, sometimes a few more, that kind of wing out in a sort of a big wide V. And then all the sub-branches that come off of it, they're all about the same length. And you think that that's not such a big deal, but then what that means is the inner branches, which would be the youngest or the oldest branches, they kind of stop growing. Oh. So as the lateral branches grow out, then you get new ones that stop growing, so that there's uniformity of the branchlets all along the stem there. A lot of the other Calyptrophora, you can see the inner ones just keep growing. Bearing. And so in the center, they're the tallest, and out toward the edges, they're the smallest because they're the newest ones. Yeah. So it's pretty interesting. And what's that?
Do you think we'll see more? I don't know if we got still shots on that one. The Calyptrophora angularis. You think we'll see more?
Let's see. Go ahead and bring up a picture of that for Kainalu and you guys to take a look at. If I can find my corals.
So one that looks different than the others? Which one is not like the others? Zoom in there, please.
Let's do a zoom on this. Aha. Okay, so that's a bamboo on the right, and probably a different species than one we've seen. And on the left is a paragorgid or bubblegum coral that has its characteristic zoanthid overgrowing a lot of the branches, and that's the yellow. So the paragorgia is the red, and that's what's actually attached to the substrate. And then the yellow is this sort of colonial zoanthid that's associated very closely with it. And all I can think of is part of the name, it's Emily Acacia something or other, because this is named by an expert for the Amy Bacco Taylor's kids, of all things, so kind of interesting.
So this little, on the right of the lasers, right on the little pointy bit of that rock, you see the V there? How do we know that that one's similar to the one we sampled, or similar to the one with all the, I don't know, the one we just saw with all the linear looking branches and just hasn't grown them yet?
Well, one of the things is that's branched too close to the base.
Okay.
I think that's one of the clues. And what Scott was looking for is something that sort of starts off as a single branch for a while, and then it starts sub-branching after that a little bit, but not very much.
Okay.
So can we just do a quick zoom on that sponge? That's just.
Yeah, sure. Go ahead, zoom.
Well, there's a lot of different animals.
Yeah. Oh, that's beautiful.
Whoa.
It looks like a donut.
Yeah, a little bit closer, yeah. Well, float my boat.
Uh-oh.
Well, we're not going to stop and collect this, but that's unusual. We've done a lot of collections. That is
come wide.
That's a weirdie.
But you don't want it?
Yeah.
You don't want it?
You really don't want it? Do you want it?
How quickly would it take to grab it?
Probably a little while.
Yeah, let's pass. Oh, this hurts.
We'll see another one just up here, don't worry.
Because this is such a fantastic community we really need. And there's an I-4, that bamboo that looks like a candelabra.
Oh, yeah.
Oh, my God. We've got so much diversity here. It's unreal. So I think that's what it is, what it's called.
Beautiful.
Very distinctive bamboo with this shape. And there's a different one, bamboo behind it. There's all types of different species of bamboo here. Scott is typing like mad right now because we're right in the community that he loves.
There's a sponge for you with a shrimp living in it, possibly.
Where is that?
Zoom in, please.
Yeah.
Oh, it's right there.
Oh, yeah.
See, they were translucent.
Is that one of those marriage?
Oh, look at that.
No,
that's beautiful.
This is Walteria, and this is Walteria leucardi, or something close to it. I'm looking at the round holes on the side of it.
Yeah,
that's one of the diagnostic features. And this is, I wanted to tell a quick story about this. I got an email this morning from a viewer, Lydia Davis, who was when we mentioned yesterday that we didn't know the origin of the name Walteria, she took the time to actually go look this up. And this was named by a researcher, Dr. Schultz, way back in the turn of the century. And he was honoring a cell biologist, Walter Fleming. So the very first species that was described for this genus, he named as Walteria flemingi. And Walter Fleming was a very sort of famous cell biologist. So Lydia, thank you very much. I already responded to her. And that was terrific that she took the time to help us with that, the origin of the name.
Coming up to a beautiful fan here.
So what is
oh, this is possibly the densest community that we've come to so far.
Yeah, definitely most diverse.
Do a snap zoom on this if it's possible.
Go ahead and zoom, please. That's dense.
Aha, so this is potentially another genus of primnoid. This could be in a genus that's called Paracalyptrophora. And I'm trying to see diagnostic characters, but typically they have smaller polyps and they form these beautiful fans with a lot of commensals on them. And I'm hoping that I'm going to get some help from Scott here. He also noted the I-4 that we found, so that's great. He said he may have seen an Acanella weberi a little while back, and I didn't see it because there's so many corals around there. He homed in on one, and if we see another one of those, we'll get a close-up of it.
Another one of which, sorry?
It's just another species of bamboo that he saw and I didn't see.
Okay.
And I'm just saying that we'll try to get a close-up to confirm that it's here. We don't need to sample that. We know what it is, but...
I'm getting a little off course here, but there's a nice bright-looking sponge.
Okay.
A lot more sponges over here you can see in Argus view.
Oh, yeah. What a place. All right.
It is.
Managed to pick a decent one this time.
Yeah, this is great.
Whoa.
This is Poliopogon, one of these big massive sponges. We took a smaller sample or a smaller specimen yesterday.
Are you thinking you're going to want to sample in the next little bit, or should we keep ship moves going?
Yeah, keep ship moves because we really need to get an idea what this community is like for a while and just run uphill. I don't think it's going to stop anytime soon. I think it's going to continue.
Don't jinx it now.
All right. Okay. Sorry. I won't say anything
Look
more.
at that sponge growing on that sponge.
Growing on a sponge.
Yeah.
Are those the same species?
Not necessarily.
Zoom in on there, please. Oh, no, it's just beside. Never mind.
Yeah, but they do that, Pilot. They really do. They will grow on top of each other. In this case, he's next to him. That's
cool. Okay.
It's
wide,
mom
please.
and her little fawn, I guess. Maybe not, but I don't know if mom kind of turns a little more yellow when she's a little bit older. Wow, look at this. We found another one of the... Oh, what is this guy?
On the left there?
Holy guacamole.
It's a big boy.
That's another one of these big pharyngimatid sponges and a lot of unbranched bamboos next to it.
Got some serious current flow here.
Yeah.
See the wiggly bamboos.
It looks so cool.
And they're all flowing from west to... No, east to west?
East to west.
East to west.
I'm 66% lateral to the left right now.
Oh, wow. Look at that. That's incredible.
This is so pretty.
This is the community, the type of community we have been looking for. These are so important, and we found one like this.
Look on Argus's screen.
I am in seventh heaven right now.
What's sixth heaven like?
I don't know.